So our final speaker today is Professor Alamelu Sundaresan. Okay, thank you. Stand corrected. And wonderful paper, I'm really looking forward to this one as well, which will be on lunar dust, environmental exposure, space, the moon, and different stuff there.
Thank you very much. I am going to take a slightly different tack on this one, since I talked a lot about the moon. I'm over the moon, but I'd rather talk a little bit about gravity in the cell, because people were quite piqued with the idea of space, cells in space, humans in space. So, and I had to go back to Shakespeare a little bit. So I said, gravity in the cell, to be or not to be, right? So one of the things that we always think about is, and I should go to the next slide, is about the organism's adaptation to the environment. So, gravity's sort of been there, done that for about 4.8 billion years. And life as we know it has always been under the influence of gravity. And again, there's many a slip between the cup and the lip. And why is that? Gravity is not your old shoe. Don't take me for granted. This was a time when I used to experiment with animations. So please bear with me. So, but it's important because gravity as a force is not tremendously strong, but it's more the fact that the sphere of it, right? The sphere of it is so all-encompassing that life's sort of just been totally, succumbing to it in a lot of different ways. It doesn't know how to react otherwise. So this is a picture or a cartoon of an astronaut, and you see the tether, and I know there are a few exercise physiologists in the room as well. One of the things when they go up in space, and I'll be talking about what happens to the human body a little bit, just so everybody can get familiar, he's actually tethered to his seat, and when they exercise as well, they have to be tethered, otherwise they would freely float. So that's the so again, gravity hurts. It hurts good, but you can feel it hoisting a loaded backpack or pushing a bike up a hill, but lack of gravity hurts too. When astronauts return from long-term stints in space, they sometimes need to be carried away in stretchers. You'll see the gait just sort of, they've lost about 2 to 5% of their bone and muscle, depending on the length of time they've been in space, and their gait completely changes. They have to get retrained to be able to move, and the muscles have to come back. And they do. It takes some time. And then I'll tell you something else about that. It's not just a force, it's a signal. Like I said, cells talk to each other, gravity is a signal. If it's not there, they talk to each other, but differently, and they might be telling each other to die. Some of them, like in a tumor cell, we've seen that it's the exact opposite. They can get more virulent. Bacteria get more virulent. So there's increased microbial virulence, which is also a risk. It tells muscles and bones, which are mechanically stimulated tissue, right? You have to give them resistance, right? It tells muscles and bones how strong they must be. When you go to 0G, muscles think they're not needed. They atrophy quickly because the body perceives there's no need for them. And the muscles used to fight gravity using the resistance, like those in the calves and spine, which maintain posture, for example. You could lose 20% of your muscle mass in a month. It's possible, depending on the conditioning level that you're at. Most of the time it's about, because astronauts are very in the peak of physical condition, it's about 5% a week. Now, these are some of the other changes that happen, okay? If you look at the blood, what happens? There's decreased immune functions because T cell signaling goes down. B cells, and that's one of the new projects that I'm on, don't make as many antibodies because their hypermutation rates go down. There's decreased RBC. You can't take EPO for that. And EPO is erythropoietin, by the way. And there's also, when you go up there, there's a headward fluid shift. It's called the cephalid fluid shift. There's increased blood volume in the head and trunk. And there's a lot of systems that get affected by that. You have neurovestibular disturbances because your otoliths, not your otoliths, but basically your little, they're like little crystals on the ear, your vestibular system gets awry. There's also the heart, there's an increased load on the heart because all the fluid. So you have about what, 7 liters of fluid in your body, at least 7 liters of blood at least, and there's some other lymphatic fluid and some other things. So imagine everything going up from down below. And the muscular deconditioning is very bad as well. Like we just talked about it, and then there's a lot of bone demineralization. You see calcium in the urine, leaches a lot. And there's also, and then the urine production goes up. So this is not just it, because there's also radiation, space radiation, this galactic cosmic rays and other high linear energy transfer types, which is more severe than what you actually see here on Earth. So you can see the before and after. So this is all 1G on the left, and you can see when they go up in space, that's Joachim Kai, that's Peggy Whitson, and I don't know the other one, but I know these two. And you can see what happens, right? You can see the head swell up. You can see the swelling on, in the face. So these are the environmental stresses of microgravity, the physical stress of flight, weightlessness. Of course, students also have this stress, right? They have academic stress, and we sometimes have used that as a model as well. And then there's confinement. So there's psychological issues as well, because you and your not so good friend are stuck up there, can't go to a movie, right? There's a lot of other issues as well. So there's, and also your cortisol goes up, your stress hormone, it goes up. And there's restricted personal hygiene, increased microbiota, okay? And that's also been characterized a lot. You were talking about the gut microbiota, and now they've actually, in the twin study, they actually investigated the microbiome of Mark Kelly and Scott Kelly. One was in the ground, and one went up there. So there's some data that they have published now. And so there's obviously, and actually they've actually documented it, although they won't release the records, increased susceptibility to opportunistic infections. And it has happened, but you'll never know. So, and then solar radiation that I just talked about, and that also is synergistic with immune suppression. So radiation is almost synergistic with a lot of different things to exacerbate the issue. So you can see the solar wind, solar flares, galactic cosmic rays, and there's a lot of factors. And so why do I study spaceflight immunology? Well, I like to look at the immune system, because the immune system has a bearing on Earth as well, because we all are always talking about resistance to disease. How do we get better public health? It all has to do with a robust immune system. Some of the things we see in space are decreases in leukocyte blastogenesis. They are a type of white blood cell. Cytokine production goes down. Inflammatory cytokine goes up, like IL-6, IL-4, etc. But the good ones go down. The subsets of the leukocytes also change in their setting. The skin test reactivity is lost a little bit, so the recall activity of the memory immune cells is also lost. Reduction in natural killer cell function, reduction in cellular migration, which I did a couple of experiments on the space shuttle. Increased oxidative stress, so free radical production. So how many of you have used a little bleach on your face to lighten your skin or whatever? Well, they use it when you do your facial, and that's how they actually, they create oxidative stress to actually do that. There's a peroxyl and peroxyl radicals that are generated, and they're quite dangerous when they're inside in your system. And then there's also changes in the expression of cell surface receptors. So when your ligand has to bind to the receptor is not expressed enough, then you don't have that pathway go on. And then the cytoskeleton, I think I talked a lot about this morning, and also cell cycle, going from one phase to another is affected. Obviously, all of this taken together will affect these space flights. And now you have the ISS, and then you have the moon and Mars. The moon has to be a step before Mars. It's a stepwise process. Now, can all of us send experiments to space? Can all of us have access to astronaut samples? No. That's a big no, right? So we use models to study things where we can do proof of concept experiments here on the ground. And so we use analogs. They have human analogs. I haven't talked about those here, but you have bed rests where the people are, they're usually volunteers, 90 days of head down, head down tilt. And it mimics a lot of the aspects of microgravity. And there's one facility in Toulouse. I don't know if the UK has one or not, I'll have to check. So I talked about the parabolic flight. You can see all the different parabolas there. There's hyper-G, and then you have a micro-G in the middle. And then you also have the sounding rockets, which go up. So if it's short-term microgravity that's generated, that you can do certain types of experiments, like early genes that can get turned on, for example, in cell. And then you also have this drop tower. There's one in Bremen that's not too far away, that's in Germany. You can have 10 seconds of analog microgravity, and you can use it only for very, very specific experiments, but it's cheap. It's not expensive. That's another thing we've got to bear. When we do experiments, we researchers, I think of us as professional beggars, because we have a hypothesis, we're trying to sell it in a way, we probably think it's the most important thing on the planet, but then the reviewer has to think that as well. So it can be pretty costly as well. And then you see that little mouse in the middle hanging by his tail. And that's a model of fine limb suspension. And that's a good model for analog microgravity as well. It's an in vivo model. So in vitro is outside the body and in vivo is inside the body. And believe it or not, these mice reproduce, they do everything in that state. They do not actually, nothing happens to them, they're not stressed. So, so the only stress is when they actually get transported from where they come to the lab, and then we give them a week to acclimatize. We do about 1 week or 2 weeks. We've done about 4 weeks of microgravity where we've, and this is a very good model because it has a neuroendocrine stress. So and those are the ones I showed you, the bioreactors. I haven't talked about them fully yet. So those are, if you look at the cylinder, okay, it's a cylinder. So there's one axis on which it's actually fixed up, right? And then it rotates on that axis. So it's a two-dimensional clinostat. So let's see what it does. So these are the different models that I just talked about. So this is what happens to cell in that bioreactor. So if you, okay, were a little cell, so those green dots inside, and the middle part is the shaft. It's a full head of cylinder. It is rotating. The nice part is the cells are in free fall, just like the astronauts are on the ISS, but the shear forces are minimal. It's only 0.2 to about 0.3 dynes per centimeter squared, and the suspension is possible. And then what happens is do you see them start coming together, right? So if you were on the ground, you sculptured some cells like skin cells, for example, they would start in 5 minutes, slowly come down. At the end of a few hours, you would start to see them form those monolayers. Look at the 1G cell culture one. And then in micro-G, you'll see them start to form little tissue-like assemblies. Do you see a bunch of clump of cells? So by serendipity, it sort of also affords us to be able to use it as a tissue engineering tool as well. So this is what a cell would look like. People were asking me what happens to the cell. In 1G, this is a lymphocyte. You see the nucleus, the DNA, etc. That's a receptor sticking out there. And this is actually based on a model of a lymphocyte from space, from the ISS. Okay, so here you can see how fluid it looks, right? We did the diagram based on the actual morphology of the cell. It can interact, it can move, it's sort of amoeboid in nature, and you see in micro-G, do you see how rigid it becomes? So there's changes in fluid distribution, gene expression, signaling. Differentiation is when it, it can actually, so you have a stem cell differentiate into something else that's functional, that's terminal. That also goes down. It doesn't move on type 1 collagen like it does in the interstitium. And also there's reduction in cell-to-cell interactions. So all of this really happens. And also with the lung cells, right? We're talking about breath. Breath, olfaction, all of this has to do with, so this could be a really good tool to look at what happens to some of the, you can culture any type of cell. And if you don't, you can culture them if they need like an anchor, like most cells, right? Blood cells are suspension cells, so you can float. They're in liquid culture or aquaculture. But the other cells all need a matrix of some sort. You could design some scaffolds, and then you could put them in there and make a construct and then study whatever you want in there. So right now, even though we can't go to the moon or do in situ moon work, we could use the moon analog and then try to culture the respiratory. We actually are trying to start to do that. So again, Mars is a future homestead. So what sort of things could happen? One of the things that could happen is humans go up there, humans reproduce and register their domain names, right? So, I mean, some of the things that could come out of this already, we are using some of the patterns for tissue regeneration, like bone cells, like we have 3 patterns on bone cells that we've been able to make bone tissue outside the human body using this particular bioreactor. And you could use it to, for example, spinal fractures, smaller fractures where you could, use the person's own cells. So you could do like tailored therapy for something like that. The same thing for lung fibrosis, could attempt to do that. So there's a lot of useful things that can actually come out of this. Then you could use it as a physiological test bed to test different drugs and see what happens. Like if you were looking at what is going to happen, is there an extreme reaction? You could just put it on the cells and have the drug absorbed in the cells and then test it and see whether, it is true or not, if there is any contention. So there's lots of different uses for this, but I just thought I'd do something different to give you a little bit of a heads up on what really happens to cells in space, because then you connect it to the rest of the body. And I showed you the body stuff early on. So right now they also have a vision impairment risk that they've introduced. And also a sort of it's called SANS, which is like a neuro, so they sort of black out sometimes. So that's another new risk that they have. So again, if there's problems in blackouts near the pons area, which is your respiratory center, then there is, your breathing, is also, because those particular centers that, from the brain that actually control breathing, heart rate, etc., is the medulla oblongata and the pons. So, which is like a little bulb down there. So there's a lot we can do with this technology, and I think more, and I think they're expanding that now. It's not just all about testing, your hypothesis up in space, but it also has to tie in with the risks that they're trying to fill in the gaps for to make astronaut travel safer. So there's a lot to discuss.